This is an OCR A-level biology video on Unit 5.5. It's the final unit of Unit 5. I'm splitting this into 3 videos for plant responses, then we're looking at the human nervous system, and then I'll do a final video which will be looking at the action of muscles. But yeah, this is the final for Unit 5, so it will actually be the final video for the first test, so then I've made all of Unit 2, all of Unit 3, all of Unit 5 to this point. I'm taking the exam in about a week's time. I think it's a week today. So the amount of videos I'm going to be producing now are going to significantly lower. I still want to finish off the course. I might try and finish off Unit 4 this week, but probably not any of Unit 6 until after that first exam, because obviously I want to narrow my attention down to Unit 2, 3 and 5. But anyway, let's get into it. Obviously some people won't really care about that, I just want to see this. So let's just start this video on plant responses. So plant responses, they have to respond to biotic factors and abiotic factors. So biotic factors are living factors and abiotic factors are non-living components. So there's various stimuli that plants need to respond to. And some examples of these stimuli, the first one being temperature. So what I'm going to do, literally list out all your things. So there's temperature, gravity, light, chemicals, touch, wind, herbivores. Okay, so they're all the ones I'm going to speak to. There's obviously going to be more. So response to temperatures, we call it a thermotropism, so like curling leaves when cold. So there's also this change that in high temperatures they may deposit thick waxy layers to help prevent too much transpiration occurring, so to keep retention of water. So in response to gravity you can have geotropisms, such as the roots grow downwards towards gravity. Light, you have phototropisms, so growing towards the light. Chemicals, we call these chemotropisms. An example of this is pollen tubes in the flower grow down the style, attracted towards the ovary. And then we've got thigmotropisms for touch. And thigmotropisms are when we've got such as like climbing plants, so like ivy wind around solid objects to gain support. Wind is not a type of tropism to wind, but you can lignify the vascular tissue, provide strength and support, so anchorage really, so the plant doesn't fall over. And in response to herbivores, there's 3 things here that they've put down. Tannins, which are found in the upper epidermis of the leaf to make them taste bad, prevent infiltration by pathogenic microorganisms in roots, and they're toxic. Alkaloids give a bitter taste to growing tips and flowers, so nothing wants to eat them. And pheromones are chemicals released by one plant to affect the behaviour or physiology of another plant. So it's sort of like a communication between different plants. As you can see here, I've got this like maroon colour, and these are looking at our tropisms. So you can have tropisms that are positive tropisms, so growing towards the stimulus, such as growing towards the light in a phototropism, that would be a positive phototropism. Or you can have a negative tropism, and that will be growing away from the stimulus. You can have non-directional responses, which we call thigmonasty responses or nastic responses, and that example here is in thermotropism, curling the leaves when cold. That is a non-directional nastic response. It doesn't grow towards tropism, it doesn't grow away, it is just a response. So thigmonasty is a nastic response as well. This chapter goes into lots of details of lots of plant hormones. But plant hormones are chemical messengers. They can be transported to a variety of tissues. They reach target cells and bind to their receptors, which are complementary to them and only act on the specific target cells. So they can influence cell division, elongation and differentiation. And this table does that I'm about to put on here does a little summary. I don't do a table on here actually, I do little bubbles, but each one's a little summary of each hormone. But obviously there's much more detail that we look at them in later in the chapter. So cytokinins influence cell division, delay leaf senescence, overcome apical dominance, and cause cell expansion. Then we've got abscisic acid, and that inhibits seed germination / growth. It causes stomatal closure when there's low water availability. Then we've got auxins, and they cause cell elongation. They inhibit side shoot growth or lateral bud growth, and also inhibit leaf abscission. We've got gibberellins, which promote seed germination and growth of stems. And finally, there's ethene, which promotes fruit ripening. Once again, as I said, we'll see those things come up much later in the video. All sorts of aspects and areas they come in. So one final thing about these hormones is a little summary. Hormone movement can be through active transport, diffusion, mass flow in the phloem and sap, and the xylem vessels. Okay, they're the three sort of methods we can move them around, and I'll write them down here. Let's look at auxins in a lot more detail. So, as we know, auxins regulate our plant growth. If you break the shoot tip apex, side branches which were dormant begin to form, and that's because we've got this thing called apical dominance, where auxin is at the top of the stem and it causes apical dominance, so no side branches can grow, because as we know, it inhibits lateral bud growth. So we've got two hormones that work directly with auxin. These are abscisic acid and cytokinins, which we've already seen. So abscisic acid also inhibits bud growth in lateral buds. When the tip is cut, auxin is removed, abscisic acid levels drop and the bud starts to grow. Okay, so direct correlation between those two. Cytokinins promote bud growth, and cytokinin can override the apical dominance, so inhibition of lateral buds, if it's directly applied to the lateral buds. So high levels of cytokinins can make the apex a sink for cytokinins produced in the roots, making growth upwards in the shoot. When the shoot tip is cut and the auxin is removed, cytokinins spread evenly around the plant and promote lateral bud growth. So we've got these two forms. We've got abscisic acid, which inhibits bud growth. High auxin levels lead to high abscisic acid levels. So if we cut the apex of the shoot, we get low auxin, then we get low abscisic acid, and then we get less inhibition of bud growth. When there's auxin present, apex is a sink for cytokinins, so they don't spread around the plant. When auxin is not present, cytokinins are free to spread around the plant and cause this lateral bud elongation. And once again, I had a little gap and I put a picture of ivy on the wall, and that's ivy growing around the wall, and that is a thigmotropism. So now let's look at gibberellins. So gibberellins have two main roles for stem elongation and seed germination. So for stem elongation, a fungal disease made rice plants grow very tall, and these fungal compounds that made this happen are called gibberellins, and these include gibberellic acid, GA3. When GA3 is applied to dwarf plants, only some can grow, and this is due to a different gibberellin member, which we call GA1. So tall pea plants, which have a dominant, just one dominant allele, so big LE or big LE, as long as they've got one big LE, which sounds really weird when I'm saying it, it'll make more sense once you see it. They have high levels of GA1. So tall pea plants, high levels of GA1. Dwarf pea plants, which have recessive alleles, have low levels of GA1. So obviously the scientific sort of hypothesis you can make from that is GA1 causes growth in stems, so stem elongation. So to prove this, they had to, obviously you can make these hypotheses, but you have to have some way to prove it. So to do that, they had to undergo this sort of experiment. So obviously the dominant gene converted GA20 to GA1, but a dwarf, which are later cross out dwarf and replaced with mutant, a completely different plant that we've not seen. It's not one of these dwarf tree plants. So a mutant tree plant is 1 centimetre tall, which did not produce any gibberellin because something earlier in the metabolic process doesn't even allow the first gibberellin molecule to be produced. So this mutant, ignore dwarf, pretend it says mutant, I do write it on later. This mutant 1 centimetre tall plant produces no gibberellin, hence it's so short. Its shoot is grafted onto the recessive plant, and when I say graft, I don't mean trying to graft a girl in a nightclub or being grafting out some hard revision. It's a technique to get a shoot onto a new plant. So this shoot from the mutant plant, which is 1 centimetre tall, is then grafted onto this recessive plant, which cannot convert GA20 to GA1. There I write on mutant. So the mutant shoot grafted onto that plant, and what they see, this mutant shoot still had this enzyme GA20, GA1. It just couldn't produce the original GA in the metabolic sequence. And they found that this shoot still grew, even though it's never responded to gibberellin before. But they proved that GA1 caused this growth, as it had this enzyme to convert GA20 to GA1, which the recessive plant didn't. So it used that enzyme and used the GA20 from the dwarf plant it was grafted onto and converted that GA20 to GA1, hence the shoot grew tall. And that is something really complicated to understand, and I was debating whether I actually even need to know it. I looked in the specification, it's not really that clear, and it does say you have to know the scientific method of how this was sort of how they found out gibberellins cause stem elongation. So my guess is you probably should learn it, look at a different source in case I didn't explain it too well. So the second thing that I need to mention here about cell growth and elongation, gibberellins cause growth and elongation, growth by elongation, and cell division. Okay, so mitosis and cell elongation causes growth. So when we're talking about growth, we're not just talking about mitosis, or we're not just talking about elongation, we're talking about both. So we also, the gibberellins promote seed germination. Seeds have gibberellin contained within them, however it is not released until the embryo which contains it releases it, and that happens when water is moving into the leaf. So in the high presence of water, gibberellin is then released. It migrates to the alurone layer within the endosperm region of the cell and causes starch production. So when we get this starch production, starch is converted to glucose, and this allows both respiration and protein synthesis. So that was all you need to know on gibberellins. Well, there's actually some more later. It's an absolutely mental topic, really big topic. But now let's look at plant cell growth. So cell walls limit the ability to divide. Growth only occurs in meristems. And this is a diagram taken from my notes just to save a bit of time for myself. And it's showing you the different meristems within a plant. So here we've got our apical meristem, and that is at the tips of stems, roots and shoots. We've got our lateral bud meristem, giving rise to side shoots. We've got our these lateral meristems responsible for shoots getting wider, giving more girth to the shoot. And we've also got these intercalary meristems located between nodes where leaves and buds branch off. And these are our intercalary meristems in green. That's all you really need to know about diagram. I've put another one here, but obviously here it's going to be really small for you. Just maybe Google meristems on a plant, and you'll be able to get more. If this diagram is not good enough for you, you'll probably be able to see what I'm trying to draw here on Google. Just do that little Google if you don't really understand it, or just look in your textbook. Right, so let's move this on to phototropic and geotropic responses, and once again we're highlighting a lot of experiments in this topic. And first of all, we're going to look at phototropic in red. So phototropic response, as we know, is a response to light. So we have 10 control plants, 10 replicates of a control plant, and we illuminate this control plant from all sides. Then we have an experimental plant, and obviously we can have lots of replicas of that, and we illuminate that from one side. Each plant stem is marked every 2 millimeters, so we get marks on our stems every 2 millimeters. This is our control plant with light shining it from all sides, so these yellow arrows indicating where the light's coming from. And our experimental plant has light just on shining from the left hand side. Let's see what happens to our plants. So this plant with light from all sides, the control plant, remains straight, nothing changes, but our experiment plant with the light shining from the left, it bends towards the left, and we can use standard deviation of the marks on the light hand side compared to the marks on the dark to see how much of a phototropic response we had. Okay, so let's have a look at the geotropic response. So we've got one control plant with, well, obviously replicas of this. We won't just carry out at once. It is lightly spun in a clinostat to ensure gravity is applied equally to all sides of the plant, and we have an experimental plant where gravity only applies to one side. In the control plant, both root and shoot grow horizontally. In the experimental plant, the upper root elongates, so the root bends downwards, and the shoot bends upwards as the lower side elongates more than the upper side. Finally, outspoken, the text for once. I found it really hard to get the right balance of speaking and getting the text moving at the right time. So then we've got these two gentlemen, Darwin, which you hope everyone recognise the name of, and then we've got Boyson and Jensen. Hopefully I've not mugged their names right off there, but they both did a very similar experiment and they got different results. Well, the results support each other, but they were looking for similar things. So Darwin proved that shoot tips are responsible for the phototropic response. So apical dominance is the cause of our phototropic response. Boyson and Jensen found that water solute movement is needed for our response. So in their experiment they had a block of gelatin lodged within our plant, and this was a permeable block, and it allowed this bending, so water and solutes could still move down. And then they did the same, but with a impermeable mica plate, and in that no solutes could move down and got no bending. As we know, auxin movement is really essential to our bending, and I'll illustrate that even more and how auxin works later. There's also this experiment on agar. So agar blocks with auxin impregnated give the same result. So different concentrations of auxin can be used to prove it is proportional to shoot curvature. So the more auxin, we'd say we'd have more shoot curvature. Up to a certain extent, both shoots and roots have an optimal auxin concentration, and you can once again Google graphs that or look in your textbook to find out your optimums, because it's not something I put in this video. So auxin always migrates from the apex to the shaded regions. The non-shaded side without the auxin tends to elongate quicker. Okay, well, not tends to, it always does, because we've got auxin inhibiting the shading side's growth, but the non-shaded side has less auxin, so it's allowed to grow. And we call the cells that are elongating the zones of elongation. So auxin travels to cells in zones of elongation, causing them to elongate. that's a key word, and sadly I didn't write it down. So zones of elongation, be thinking about that when you're thinking about how auxin works, which we'll look at now. So auxin promotes hydrogen ions active transport by ATPase on plasma membranes towards the cell wall. So we're pumping hydrogen ions towards the cell wall. This obviously produces a low pH, which is optimal for our wall loosening enzymes. These wall loosening enzymes break bonds within the cellulose, while hydrogen ions disrupt hydrogen bonds in the cellulose. And we know about the cellulose structure from unit 2.2, whatever unit biological molecules is. You can have a look, the video's out there. So we're disrupting these hydrogen bonds between cellulose molecules. We make the wall even more permeable, it becomes less rigid, and the cell takes up more water. Therefore, if the cell's taking up more water, it's going to swell, the cell elongates, the wall loosening enzymes work at the top and bottom of the cell, so we see it elongate upwards and not outwards. Phototropin 1 is an enzyme which increases reaction in response to blue light. The active phototropin 1 gradient causes the movement of auxin from light to shaded cells. So the study of that is not actually that in depth. I believe they're still not quite sure how things like this occur. It's still sort of a hypothesis, but one thing that is known, phototropin 1 causes this movement, and that's an enzyme. And that is, in a nutshell, how auxin works. In geotropisms, auxin settles out on root ends due to gravity and has the opposite effect in the stem. Therefore, root ends do not elongate, the starts of our roots do. And to be honest, if that wasn't a solid explanation for you, I understand, because for me it's not a solid explanation. You might want to do a little bit more background reading into how auxin works in the roots. Obviously I'm a student, not a teacher, so if you think I say something that you don't understand or think I'm wrong, please challenge it, especially if you're watching it now, or just check a different source out, because I'm not a qualified teacher. Finally, in this topic, we're going to look at the commercial use of plant hormones. So we've got auxins, which we've been speaking about the whole video, and they can promote flowering, prevent leaf and fruit drop. We can use cuttings can use auxin, so you dip the end in cutting rooting powder to encourage root growth. Auxins are used in seedless fruit production, so unpollinated plants can be treated with auxin as it promotes ovule growth, which triggers automatic production of auxin in developing seedless fruits. Herbicides, they use auxins to kill weeds, promote shoot growth so much that the plant can no longer support itself and it dies, which I think is really interesting. But obviously there's issues with herbicides. You can apply it to the weed, and sometimes the auxin can kill other plants as well. But I don't think all herbicides use auxin. There's different forms, obviously. So cytokinins delay leaf senescence, prevent yellowing of lettuce leaves. They're used in tissue culture, so they promote bud and shoot growth, and they also promote plants with lots of side branches. So we use cytokinins. We can sort of manipulate how we want plants to look for aesthetic reasons usually. It's not the most popular hormone to use, but it's definitely good for this, preventing our yellowing of lettuce leaves, which I think everyone finds an issue. Tissue culture, obviously, all these hormones are used at various points of tissue culture and promote bud and shoot growth, to produce new plants with lots of side branches. And that's the thing, cytokinins, as we remember, promote side branches. So if we apply cytokinins, we can sort of get bushy products. Okay, bushy plants rather than tall, long, thin plants. So gibberellins are used in many things, one being fruit production, so they delay senescence in citrus fruit, extending the picking time. Gibberellins work with cytokinins to make fruit elongate. Grape stalks elongate and the amount of size of grapes increase, so gibberellins are really good for fruit production. They're also used in brewing, so beer is produced in the malt house, and when barley seeds germinate, the alurone layer of the seed produces amylase enzymes that break down stored starch into maltose. Addition of gibberellins speed up amylase production. Then we've got sugar production. Spraying sugarcane with gibberellins stimulates growth between the nodes, so we can get stem elongation, increase sugar in the internodes. Okay, so sugar in production, and finally for gibberellins we can use plant breeding. Conifer plants have a really long juvenile stage of growth, and we can use gibberellins to speed up that juvenile stage of growth, induce seed formation. And another thing we can do is use gibberellin inhibitors. So this is not gibberellins basically, they can help produce desired characteristics such as short stocky flowers, prevent the lodging of stems. So similar to the use of cytokinins, gibberellin inhibitors produce these short stocky features, and they can also be used to prevent stems basically bending over, and that's what we say when we are talking about lodging. Then we've finally got ethene to end up this horrible video at the start of unit 5.5, and I've got a hard word to announce here, and sorry in advance for this pronunciation. Chloroethyllophosphoric acid. So chloroethyllophosphoric acid produced to put ethene in solution. I think I had a good attempt at that word. They speed up fruit ripening, promote fruit drop, promote female sex expression and lateral growth. Ethene inhibition helps transport of plants, such as bananas, to prevent over-ripening. So this again, I didn't write here, but while we use ethene in commercial use, we're actually more often using ethene inhibition to prevent over-ripening, because we pick bananas abroad, and if we had bananas that were getting overripe, it would be no good, because you've got to transport them from Africa, and by the time we got them over to the UK they'd be too ripe. So we have to put ethene inhibitors to prevent them over-ripening, and then we actually spray them with ethene when they get into the country to make sure they ripen again. Otherwise we'll just have green bananas, and some people, depends what you fancy really. But that is the 1/3 of unit 5.5, and I probably underestimated how big this topic is. It's about the size of some units put together, and it's a really weird topic. It's putting plants, nervous system and muscles together, and I think OCR had these 3 topics left, and they were like, Jesus, what are we going to do with these? Let's shove them all in one. Anyway, some more videos to come. Hopefully get this topic done by tomorrow. I know many of you are not watching these every day, but never know who's going to be watching this video. Anyway, see you next time.